Go ahead and get started. So as you saw in Alyssa's talk, there's a couple citations of the work I'm presenting here. As many of you know, sometimes the field work informs a lab experiment. Sometimes the lab experiment informs the field work. In this case, we had done this study in 2019 where we sort of validated this water cortisol method for these small fish. And we've been published in other places, but it's not the same as actually doing it in your own lab. And that sort of gave us inspiration to be able to carry out Alyssa's experiment. And one of her big things, which I agree with, is let's see how we can assess the health of these fish without lethal techniques. So we developed some of that through the experiment I'm going to describe today. So I first need to acknowledge really the first author on this, but she's graduated, so I'm the presenter. Elyse Schnabel graduated in 2019 from Millikan. She's now in physician's assistant school at Thomas Jefferson University in Pennsylvania. She was a James Millikan Scholar, which meant she had extra research responsibilities as part of her degree program. And this project was chosen as the James Millikan Scholar Project of the Year in 2019. And in late 2020, we actually published this work in Fish Physiology and Biochemistry. So we're going to start with the fertilizer that we use in the study, which is ammonium sulfate. Ammonium sulfate can enter water unintentionally, of course, via runoff, but it's also sometimes used intentionally in waterways as an algicide. Of course, that's typically going to be in private ponds or waterways, not just regularly dumped into public waterways. And its primary use is as a fertilizer for alkaline soils. The ammonium ion is released and it forms a small amount of acid. So you end up with a decrease in soil pH and an increase in available nitrogen for plant growth. It's also used in other ways. So it has lots of different applications, and it includes use as an adjuvant for water-soluble pesticides. It binds iron, it binds calcium cations, and it's extra useful in the delivery of glyphosate and glufosinate to crops. And we also know that it can be responsible for sulfate deposition into waterways. Other studies have estimated that only about 20% of the sulfur deposition in central Illinois is actually environmental through natural sulfur cycles, and the rest is anthropogenic from the process of adding products like ammonium sulfate to fields. So when it comes to ammonium sulfate and its potential as a toxin to fish, the effects of acute ammonia toxicity include increased breathing, respiratory rate, oxygen uptake, and cardiac output. There are more serious effects of long-term chronic exposure to ammonia, which include nervous system breakdown, convulsions, and death. And of course, as I already mentioned, and everyone knows, sulfates are natural and essential for plant growth, but their ability to form strong acids can still cause a problem if you're applying large amounts of sulfates to land and then runoff occurs into the waterways. So Alyssa touched on this already a little bit, so I won't dwell on it too much nor bore everyone with the details of full-blown cortisol physiology here. But when we're looking for biomarkers of fish health, we know that fish can secrete a variety of hormones, including cortisol, through exocrine glands and through their epithelial cells themselves, exiting their gills and into the water that surrounds them, and typically after undergoing some kind of stressful stimulus. And also, as was previously discussed, this is not a bad thing. If there is a acute stressor, it sort of increases the metabolic activity and mobilizes glucose to the tissue so that you can sort of escape or face the stressor. But chronically, that eventually leads to the tearing down of things like protein-based tissues and fats, and also the shutting down of energetically expensive processes like immunity and reproduction so that you can continue to address the stressor. So that's one of the ways in which this is problematic if it becomes chronic. And we know that it can be measured from the blood, it can be measured from gut contents, mucus, and other studies have shown from the water. And again, long-term secretion can be problematic for a fish's health. We know that the aquatic environment tends to expose organisms to more pathogens sort of immediately and harder to escape than an aerial environment. And so innate immune defenses in fish in particular, such as lysozymes, are very important for their initial defense against pathogens. So in this study, we used western mosquitofish, or Gambusia affinis, as our model species. They are an introduced species throughout much of their range throughout the United States, which includes introductions in central and northern Illinois, but they do have a native range through southern Illinois and the southern half of Illinois. They're very small, short-lived, live-bearing fish, and we really just chose them as a model species for the study because they're easy to get and it was easy to maintain them in the lab. And of course, sadly, while we followed all protocols for animal care, unlike the redfin shiners, we were trying to return them to the wild safely. We had no interest in doing that with an invasive species. So purpose of the study was to determine if different concentrations of the fertilizer would affect their stress physiology and innate immunity. We hypothesized that more of the ammonium sulfate in the water would reduce or would increase cortisol production, and that would decrease their lysozyme or their innate immune defenses against a bacterium. So we did 2 experiments. We had the exact same laboratory setup, except for we used different numbers of fish. And because the cortisol test was non-lethal and the testing for blood samples for the lysozyme activity was lethal, we did not use the same set of fish for both. And we chose concentrations based on environmentally relevant levels. They were also about half to a quarter of the LC50 levels for rainbow trout fry. And we had our laboratory set up with these containers and aged tap water, 3-day aged tap water that was aerated for at least an hour per day. And every container had its own bubble stone. And we changed the water completely twice per week. So we did 2 extractions of cortisol along the way. So we put fish in the cup, as Alyssa just described, 14 mils of water for 30 minutes. We then pulled 6 mils of that through the filter, passed it through with methanol, and then crystallized the hormone by dehydrating the sample and reconstituted it in assay buffer. We also photographed individual fish because, as I mentioned earlier, I did not mention this, but we did have some of them in shared containers in their living conditions, in their rearing water. And so we used photography and Adobe Photoshop to make sure we were matching samples with the right fish each time. They did swim in their sample cup for 30 minutes in isolation. And then we used the same cortisol assay kit that was described previously to measure hormone levels. So for the lysozyme, the fish were anesthetized at the end of the experiment with MS-222, and we pulled 5 microliter blood samples from the fish using a Hamilton syringe and a very small needle. And we did a lysozyme assay using lysis of known concentrations of micrococcus over a 15-minute period. And we compared those sample values to a standard curve of lysozyme from chicken eggs, which is one of the most potent lysozyme samples you can find in the vertebrate world. We also did a survival analysis and ran some statistical models considering that we had shared containers and living conditions, and then we had repeated samples for the lysozyme time to control for those variables that were introduced that were not controlled for as part of the study. We will start with the survival data. And as you can see, we did have a difference in survival. We did not really anticipate this. We thought we would be sublethal on this, but as you can see in our control group, even we lost about 18 to 19% of our fish just for lab conditions, and lost a lot more along the way in the other 2 groups. So clearly these animals did not handle this as well, even as the rainbow trout fry that were used also in reference to our choice of concentrations. Here is the data for the cortisol that was log transformed because it wasn't normally distributed. You can see there's no difference at week 1 and week 2 in the control group, although cortisol trended down after longer time in the lab. We see there is a significant difference in 2 standard errors here, so that's why the bars overlap, in the 40 parts per million with higher cortisol after week 2 when exposed to 40 parts per million. And after week 2 at 80 parts per million, we also still had significantly greater cortisol, but you will see that there was no difference after 1 week of exposure. So clearly it took longer exposure than just a week to see these physiological effects. And so on an acute standpoint, there was not evidence that this was necessarily very problematic for the fish. When we look at the lysozyme activity, the one on the left shows the different time points of how much bacteria lysed at 5, 10, and 15 minutes. The one on the right shows the overall average because we didn't see any major patterns across the different time points. And what you'll see is that the ability to lyse bacteria, so basic immune defenses, were much greater at 0 and 40 parts per million and significantly reduced at the 80 parts per million group over there on the right. So our hypothesis that a higher concentration of ammonium sulfate would induce more stress in the mosquitofish was supported, as indicated by the increased cortisol levels. In addition, the mosquitofish survival did decrease after lengthy exposure to ammonium sulfate, not so much early on, nor do we see a lot of stress early on. And while we know that this species is very tolerant to poor water conditions, it may not include a heavy dose of ammonium sulfate in the water that has been exposed perhaps after a heavy rainfall, shortly after fertilization of soil. So another study similar to this, there was another study with mosquitofish looking at organic insecticides in particular, and they also found that there was no serious effects from the chemicals at a rate of 2 pounds to 1 pound per acre, which is not a very high concentration to consider that volume. But mosquitofish clearly could be exposed to lesser amounts of the fertilizer than used in our experiment with little effects. And finally, why is this sort of important, and why did it inspire some of what we did with Elyse's work? I think in my career of working as an ecologist and working with different types of vertebrates, and even my experience working for the Natural History Survey before grad school, I think that the small fish and the stream fish are often overlooked in conservation and management efforts, and at least they're de-emphasized compared to sport fish. And I get the social part of that and why we care so much about sport fish, but there are definitely conservation concerns and stories that need to come out of the work with stream fish as well. And so while it's unknown to the degree to which these costs of increased stress and decreased immunity might impact their fitness long-term in the wild, certainly even reduced survival rates of model species like the Western mosquitofish could be of great concern for these other stream-dwelling fish. So with that, I would like to thank Millikan University, my host institution here, for funding work like this and work with our students. And the Illinois State Academy of Science provided a grant to Elyse to fund the cortisol assays of this work. And here are my social media handles if you want to follow me and the work that my lab does on there.